Hey, good morning, everyone. Thank you so much for joining today's webinar on OpenCRAVAT. This is Kayla Strauss. I will be facilitating the WebEx today. Please let me know if you have any issues with the screen or audio. So this is one of a series of webinars on ITCR tools that CBIT has scheduled for the NCI staff. And we hope that these tools might be useful to your research. We wanted to let you know that we are recording this session for future reference. And during this session, if you have any questions, please just use the chat function to type your question, and we can answer your questions during the question and answer portion of the webinar at the end. After the session, let us know if you do have any additional questions, and I can certainly pass them along to our presenter. So without further ado, I would like to introduce Dr. Rachel Karchin. Thank you, Rachel, for your time today.
Well, thank you for having me. I'm going to start here by sharing my screen. Yeah, thanks to everybody for attending. And I'm going to be speaking today about OpenCRAVAT, which is a variant annotation meta annotator. So for those of you who are not familiar with me, I am probably most of you. I am a professor at Johns Hopkins, and I am a core member of the Institute for Computational Medicine at Hopkins. And I'm going to start by showing you the OpenCRAVAT mission statement. And here we go. So the motivation behind OpenCRAVAT is that we want to make the best quality interpretation of genomic variants accessible to the widest possible audience, including researchers, clinicians, and citizen scientists. And we're also very interested in reaching those who are in resource limited situations, such as in low-income countries. So unlike most variant annotation tools that are of high quality, you don't have to pay for OpenCRAVAT. It's completely open. And we have aggregated over 250 different tools that can be used for variant interpretation. With funding from the NCI, I believe we have been able to match the quality of expensive commercial packages. And we've really tried to achieve, the best type of software engineering practices. We know that many freeware tools, tools from academic labs, are often not well supported, or maybe are kind of buggy and just don't match the quality of commercial tools, which is why people often spend lots and lots of money on commercial tools. And our goal has been to get around this and to just provide really high quality tools for everybody. So here is our landing page. I encourage you to memorize this URL, go to our landing page, which provides very extensive instructions about how to get started using the tool, a lot of detail about what it provides. And at the NIH, we also have a local installation on Beowulf. So if you use Beowulf, you can access OpenCRAVAT that way. Here's a slide about our history. So we are a product of about 10 years of funding from the National Cancer Institute. In 2013, we were one of the first projects from ITCR, which is Informatics Tools for Cancer Research, which is a program project that was designed to give support to cancer researchers who are computational. And many of us struggled in the years before this to get funding to support our work, because NCI really prefers researchers to have a very strong experimental component. So wet lab research is prioritized. Computational or dry lab research was not so prioritized. And some program officers at NCI who really believed in the computational side of cancer research, launched the ITCR program. And it's grown from about 6 funded projects, I think, in 2013, to now something like 70 or 75 projects. So it's grown and provides a really wide range of valuable tools designed to support wet lab researchers. And with, again, a particular emphasis on users of computational tools who may not be computer science experts. So here is an overview of some of what we've accomplished. We began with a website called cravat.us and one called Muppet. And these websites were designed to publicize tools that were developed in my own research lab. So I had developed variant effect predictors, both oriented towards cancer and also just towards pathogenic variants in general that were disease agnostic. And we had also developed a tool that maps missense mutations onto protein structures. And again, this tool, Muppet, was designed to be usable by people who weren't experts at protein bioinformatics. In the following years, we made a number of technical advances. We started, rather than running these classifiers in real time, to pre-compute every possible position in the annotated human exome and with all possible amino acid substitutions. So using a database with a pre-compute like this, we were able to deliver variant classification results very fast. And you can see here, if you scan this slide, just some of the highlights and how we've developed technically. We did a pre-compute of all TCGA mutations in 2015. And this spanned mutations called in over 9,000 patients. All these mutations were mapped onto protein structures in Muppet. We then introduced a Docker container to make it very easy to use our tools for people who didn't want to use them on the web. And we started integrating with a lot of different other tools in ITCR. And you can see a lot of these on this slide. We have Trinity, UCSC Xena, Galaxy, BRCA Exchange, Index, Civic. So you can see through the years we have integrated with more and more ITCR tools. Then in 2019, we made a major transition from the older Cravat to OpenCRAVAT, which is a Python package with multiple endpoints. So it can be installed locally, it can be run on the web, can be run on different operating systems, it can be run in the cloud. And we started really pumping up the growth of all the types of annotation tools that were available in what we call an app store, which is makes it very easy to use different types of variant and gene annotation tools. And I'll talk about this a little bit later in the talk. And I believe this app store, it makes OpenCRAVAT unique in terms of its usability and also the breadth of the resources that it gives you access to. So in the past couple of years, we have branched into the cloud, and we're also in the process of adding a bunch of new types of annotations. And I'll also talk about this later in the talk. Okay, so the first thing I'm going to talk about is what Cravat looks like from the outside. And I'll spend a bunch of time on this. And after that, I'm going to talk a bit about what OpenCRAVAT looks like from the inside in terms of its architecture and so forth. So the really the heart of OpenCRAVAT is a browser-based graphical user interface. So this is served in a web browser, which may be on our public website, but if you're running OpenCRAVAT on your local machine, it looks exactly like the website, except that what you see in your web browser is completely private to your local machine. And there are two main tabs in this GUI, which I'm going to show you right now. One is the Jobs tab and one is the Store tab. So this is what OpenCRAVAT looks like, either in the on the website or in the GUI. So what you're looking at here is the Jobs tab. And here, this left-hand side of the screen is a controller for how you would submit your jobs through the GUI. So it accepts many different types of formats, tab-separated format, VCF formats. It also accepts formats from 23andMe, Ancestry DNA, Family Tree DNA. You can also insert lists of dbSNP, RSIDs, and coming soon, you're going to be able to submit lists of just HGVS IDs and also allele registry IDs from ClinGen. So your input can consist of one or many files. So you can put in many, many files. If you want to annotate the variants on a cohort, you can then select your genome assembly version. We support 18, 19, and 38. So if your variants are not in HG38, it's very easy to still submit them. The annotators available to you are then organized by category. So you can see the categories down here. And you can click on these buttons and you see a checklist of annotators that are available in that particular category. When you're then ready to submit your job, you just click this annotate button. Now the right side of the screen, once you click annotate, you will get a new row here in this jobs table, which will show you the state of your job as it goes through different parts of our annotation pipeline. And when it finishes, you have this option to download in various formats just by clicking on buttons here in this download column. You also here get a whole history of jobs that you've previously submitted, so you can go back and look at and reanalyze your data, re-review previous jobs. So probably what most people will do who are using the GUI is click this Open Result Viewer button. But I think before we look at that, I'm going to show you the second tab on the jobs page, and that's the store page. So at this time, we have 289 modules available. This might seem like an overwhelming number of possible annotators, but we've gone to great lengths to organize them for you. So they are all tagged here by different categories. You can arrange them by their update status. So you could put the most recently updated modules up at the top. You can rank them by popularity. You can rank them by name, by size. And again, I think very useful is these different types of categories of annotators. I'm just going to go back here for a moment. So the modules include not only annotation modules, but there are also different types of graphical widgets that you can include. Then we have input converters, which support different types of input file types. And we have what we call reporters that support output of different file types. If you want to learn more about a module, you just click on the tile and it flips around and you see documentation and a lot of metadata about that particular module. You can also install uninstall or update your modules just by clicking on buttons on the tile. And the store lives on a local web server at Johns Hopkins, and it's also mirrored in the cloud both to Amazon through their open data program and also to Microsoft Azure through what they call their genomic data lake. So there's many ways you can get to the store. All right, what I'm going to talk about next is our graphical results viewer. So when a job completes, you can open your result viewer, and you do it by clicking on this button right here. And there are 5 tabs available on the result viewer, and we're going to walk through them right now. So the first tab that opens is the summary tab. And this shows you metadata about your job. So your creation date, your input file name, your reference genome assembly, number of unique variants you submitted, and so forth. If you have submitted over 100,000 variants in the GUI, you see this red bar on the summary tab. So this is a limitation of web browser technology. You can look at only a maximum of 100,000 variants in the GUI. So to go farther for larger jobs like this, you have to move to the filter tab to narrow down your job in some way to get below that 100,000 mark. So I'm going to go through the filter tab, and first I'm going to show you that after you've filtered, the summary tab shows you some very nice summary widgets in addition to the metadata. So we have donut charts of different variant sequence ontologies, coding and non-coding variants. We show bar charts of gene ontologies and a circus plot of the locations of the variants in the job. And in fact, you can save these widgets to disk and you can use them in publications if you so desire. So the filter tab allows you to filter samples, genes, and variants. The sample and genes filters are pretty straightforward. So the samples filter gives you a checkbox of all the different samples you've submitted. So these are typically all the individuals in your cohort. The genes filter allows you to just look at results for a particular gene or genes of interest. So you would just put in a list of genes in Hugo format into this list. The variant properties filter has 2 components. So there are shortcut smart filters that allow you to limit your results with very simply to maybe a single chromosome, coding or non-coding, depending on the annotators that you have used in your job. You may restrict yourself just to variants that have annotations for those particular annotators, for example, ClinVar or Cosmic. You can also limit by sequence ontology. However, our pride and joy is this query builder, which allows you a very, very flexible, fine-grained filter design using Boolean operators. And the operators can be applied to any property of an annotator that you have used in your job. So let's take a look next at the query builder. So here is an example. That's a typo. This is an example of the query builder combined with a gene filter. So what I've done here is I've listed a couple of genes I might be interested in. So I selected BRCA1, BRCA2, and TP53. And then in my job here, I used both ClinVar and I used PhyloP, which is a variant annotator that scores variants by conservation on the nucleotide level. So what I've done here is I created a Boolean combination of variants that have a particular clinical significance value in ClinVar And this gave me a checkbox when I selected clinical significance. And so here I selected pathogenic or pathogenic risk factor. And I could have selected any combination of these values of clinical significance in ClinVar. I just selected these two as an example. And then I used the AND Boolean operator. I could also have used OR. And I looked for a phyllo-P score in vertebrate genome alignments that was greater than 20, which is something that phyllo-P advises that if your score is greater than 20, you're highly conserved. So if I click on this little icon here, this would then reduce the number of variants in my job from about 5 million to the number of variants that met these criteria. And then I would apply the filter and go on to the variant and gene pages to see annotations just of those variants. And I'm going to show one more example of how you could occupy the query builder. So here I'm using the query builder to find hotspot variants in Cosmic. So this is a pretty simple filter. I'm just saying that in Cosmic, I want variants whose variant count is greater than or equal to 5, which is a common definition of a hotspot. I could make this a lot more complicated. Again, I could limit it to certain genes. I could limit it to certain cancer types. So this query builder in combination with the sample and gene filters really allows you for very, very fine-grained filtering of your variants, which is the idea of OpenCRAVAT. We assume that you begin with an intractable number of variants and that our tools allow you to filter and sort by lots of criteria to narrow down to a few variants that would be of greatest interest to you for further study. So next, I'm going to go to the variant tab here. And here's a screenshot of the variant tab. It's split into two. And the upper half is a dynamic table, and the bottom half is widgets. You can, in fact, toggle between table only or widget only, or you can see the split view. And each row in the tabular pane is a variant, and it shows you information from all the annotators in your job. So you can see here in this job, well, I have these basic variant annotations that come with every job. And then I've used ClinVar, Cosmic, GERP++, which is another conservation metric based on DNA. Linsight here is a annotator based on another measure of nucleotide conservation, more specifically negative selection. And FastCons here is, again, another measure of nucleotide conservation. So I've highlighted this row, and as I highlight the row, widgets representing just the variant that I've highlighted come onto the pane. And then I can use my pointer to move down the rows, and the widgets that I'm seeing will change. Again, the columns can be sorted and filtered beyond your original filter. And this gives you a very dynamic way to explore the variants in your job. We also have a gene tab that aggregates the variants onto different genes of interest. You can take a look at the breakdown of coding variants and non-coding variants in your genes. You see the most severe sequence ontology in the gene. You see all the sequence ontology in the genes. We have a protein diagram. This is also available for individual variants, but it's also interesting to look at this lollipop of all the variants in your gene with their sequence ontologies, the number of samples in your job, if you have a multi-sample job, if you're annotating a cohort. Here on the top, you see the sequence annotation, the annotations of the ontologies of the variants in your job. and you can then compare to different sets of reference variants. So here I've selected reference variants from TCGA, only from breast cancer, and here you see all the variants in TCGA from breast cancer patients, and you see them here with their different sequence ontologies, and also if these variants occurred more than once, you should see a longer stem in the lollipop. Next, we have the layout tab. This is simply a way to configure your layout. So if you want to only look at, here I'm on the gene tab, you can do something similar for the variant tab. If I just want to look at particular columns, I can select them here. And I can, once I have my table configured, I can save my view, I can reload it, and I can rename it. As you'll see going further, you can also save the state of your gene and variant tabs, and then share them with others if you want to share your analysis with collaborators. Okay, so I've been talking a lot about the graphical user interface. I would like to say that we also have a command line interface. And everything you can do with the GUI, you can also do on the command line. And I'm going to spend less time discussing this because this is kind of a dry topic. But you can see here's an example of how you can run OpenCRAVAT on the command line. Here, instead of clicking that you're going to annotate your job, you would use the OC run command, give your input file and your reference genome, your input format. You would have a list of annotators you're going to run and your output format. And this is more useful for people who want to use OpenCRAVAT programmatically, typically in scripts that are part of a larger pipeline. All these command line commands are documented extensively in our manual, and we also have a quick start video for the command line. One thing that we have done that makes OpenCRAVAT very different from our original web platform is that we have multiple endpoints. So there's many ways that you can run OpenCRAVAT. So you can run it on the web, but you can also install it on your laptop, on a larger desktop or server machine. You can be running Windows, Mac OS, or Linux, and you will get exactly the same interface, the same GUI and the same command line interface that I just showed you. For Linux systems, there's a multi-user version available, and I believe this is what is installed on Beowulf. So this way you can log in through a login screen and keep all your jobs separate from everyone else who is running that local installation. Very easy to install on a local system. We have native Windows and Mac installers, and you can also use pip. Pip install OpenCRAVAT if you are a Python user. And if you don't want to install it all, you can run it in the web and also on some of the cloud platforms that I'll show you later. In addition to what I've shown you, we have some more extended analysis options. So one of them is that you can do a case control analysis if you have, again, a whole cohort and you've uploaded multiple variant files for many different samples, or a multi-sample variant file. You can then additionally add a file where you list the IDs of your everyone in your study, and you basically indicate whether they are case or control. And then you can find variants with significantly different frequencies between two groups of samples. So this is very nice. You then just, in addition to all your other annotations, you get p-values for the different frequencies of the particular variant in your cases and your controls as you've defined them. We use different inheritance models. You can use allelic, dominant, or recessive models. And then you can just see these p-values together just as another feature, another column with all the other annotations in your job. Again, you could also use filtering if you simply want to look at the variants that have a significant p-value. We have something called a single variant page report that we think is interesting to many users. And this means if you are just the exact opposite to someone who has a large cohort and millions of variants and wants to find out which of those variants are most interesting, what if you start out and you only have one variant or you have a very short list of variants? So we have a mechanism where you can submit a request for just a single variant and you get back a very nicely formatted report. You basically get information from 80 of our annotators, and this also includes visualization widgets. So here's a screenshot of this particular variant. You get to see some of the variant information that when we looked at that dynamic table, these were the columns that were on the left side of the table. And you also here you can click on any of these buttons and you can go to a section of this report that gives you a lot of information from particular selected annotators. You can also, if you have a web tool and you are interested in linking out to the single variant report pages, there's a very straightforward way that you can use a structured URL and then just populate pages in your tool and link out to this. And a number of groups have done this. For example, Civic and CEDAR, which is a web tool that shows immunogenic variants. So this is, again, a very interesting way to use OpenCRAVAT. Here is the Muppet tool, which this was one of the first tools that we developed back in 2013. And now, in addition to having its own website, you can also select this as an annotator. So this tool was designed to make useful interpretations of mutations available for users who don't have great access to expert protein bioinformaticians, but might benefit from seeing their variants on protein structures. So what's different about this tool and many others is that the protein structures have been annotated. And you can click on these annotation buttons and see various regions or positions on the protein structure where perhaps a small molecule ligand binds, or there's a known protein-protein interface. For this particular structure here, which is very famous cancer protein, you can also see where various inhibitors bind onto the structure. If you click, this thing is very sensitive. If you click onto these buttons, you can also view mutation hotspots from different, from TCGA pan cancer or from selected TCGA cancer types. So you see here these kind of red jelly-like looking regions. These are hotspots where you have increased missense mutation density compared to just a background model of the distribution of missense mutations on a protein structure. Again, you can access these structures through structured URLs. So you can include links to Muppet from bioinformatics web tools where you may want to add this kind of annotation. That is a quick tour through what you see as a user of OpenCRAVAT. I'm also going to spend just a little bit of time talking about what happens under the hood. So we have designed OpenCRAVAT to be a very robust piece of software. We didn't want to be flimsy academic ware. So I'm going to talk a little bit how we have designed the tool to run. So it is modular and extensible. You see here on the left an overview of our architecture. So we have these different converter modules, which are all independent of each other, and they're extensible. So it's very straightforward to add a new converter module. Again, we've had a lot of demand for HGVS and ClinGen allele registry ID inputs. So we'll be adding new converters for these very soon. If you have an input format that would make OpenCRAVAT a lot more useful to you, you can send us a request and we will create a new input converter module for you. Our annotators here, which you see over on the left, come from many publicly available resources. And we build these into SQLite databases. So everything is pre-computed. So by the time you look at our App Store and see these annotators and other types of modules available for download in the App Store, pretty much what you're getting is a MySQL database, and that's what you download onto your local machine if you're running it locally. So you've essentially brought the data to your analysis tool. We also have mapper modules. So again, we take data typically in genomic format. Our input representation for variants is genomic. And we do very complete mapping onto transcripts, protein sequences, and also onto protein structures. And then when a user requests a annotation from a particular module, that map data is then shown to all the annotation modules, and the annotation modules then return annotations to each mapped variant. The system then generates another SQLite database called the annotation result database, and this is returned to you. This is something you can use to reopen your job on any machine or share your job with users. And you can also use this SQLite database to generate a report in these multiple other formats. So you can get VCF format, you can get Excel format, text formats, dbSNP format, and so forth. And you can do that dynamically as soon as you have that the SQLite database. Here's what it looks like. You can, in fact, open up the SQLite database if you like. And in addition to all the information about your job, the state of your job after you've opened it and done analysis, for example, all of the things you might have configured, the filters you've applied, you know, the tables whose view you've optimized to how you like them. All this is saved in your SQLite results database. So again, this makes your research extremely reproducible by yourself or by others. One thing I haven't talked about yet is that OC is available in the cloud. And we have been working with Microsoft, Amazon, and DNA Nexus to incorporate OC into their cloud systems. So here is our GitHub for Microsoft Azure users. So you can run it in, you can run OC on the Azure platform. You set up a virtual machine, and you pull store data from their genomic data lake. And we have, if you are again, a power user who runs stuff in the cloud, and you have Azure credits, Azure expertise, you can go to our Read the Docs page and our GitHub and learn how to get OC running in Azure. And likewise for Amazon, we have created a CloudFormation template that you can use to annotate your files in an S3 bucket. We have detailed documentation of how to do this. And again, like with Microsoft, we have another copy of the store. These stores are identical and they're all synced every day with the store that is on the web servers at Hopkins. But the OC store is also available through the AWS Registry of Open Data. And finally, we have partnered with DNA Nexus for a very specialized type of cloud application. So we created an applet for extremely high throughput annotation of very large cohorts with DNA Nexus. So to handle really, really huge cohorts, like for example, what if you want to annotate the entire UK biobank? You can do this on DNA Nexus, and our applet is very simplified. You input a VCF file, and again, you select the annotators you want, and your output is then an annotated VCF file. And of course, these are just huge multi-sample VCF files. So this is an efficient way to use our system for really gigantic cohorts, which wouldn't be tractable with the type of software I've shown you so far. We have some new features under development, which I'm going to talk about. These are packages and cohort management. And we have released an initial version of packages. So I'm going to tell you a little bit about what they are. So as we have grown to have, again, almost 300 modules in the system, we realize that it can be intimidating to some people who are new users of the system. And to make this easier, to simplify a selection of annotators, filters, layouts, and reports, we have created these packages. So these are pre-selected combinations of annotators, filters, layouts, and reports. So we've got 5 of them available right now. You can use the ones that we've created. You can modify the ones that we've created, because once you've opened it, you can use the filter tab to change the filters. And in the upper right-hand corner of the Jobs tab, after you've run a job, you will see this icon that looks like a package. And you can click on this, and you can turn any job that you've run into a package that you can reuse. You can keep it privately for yourself, or if you think it's particularly good, you can publish your package to save it with our whole user base. So let me give some examples. So here again, like our other annotators, if you want to learn about a package, you click on the tile and it flips around. And here, so here's an example. I've flipped around the tile for the drug interaction package. So this identifies variants with potential impact on drug response. So we began here, it's fairly simple. It uses only 3 annotators, which are ClinVar, PharmGKB, and the drug interaction database, DGIDB. And it's filtered to show variants that only have, so this is a Boolean OR, variants that either have a ClinVar annotation of drug response, or are listed in PharmGKB, or are non-synonymous variants in genes that are listed in DG-IDB, which does gene level annotations. And it also provides a very clean layout of selected widgets and Excel and tab delimited reports. And here is, oh, okay, I think that's the one example I'm showing of a package. I'm gonna next just show cohort management. So we haven't released this yet, but we had requests of that users would like to have ways to compare groups within a large multi-sample job. So we are here providing tools that if you run a large multi-sample job and you have metadata about the samples, for example, maybe it's a cohort of cancer patients, maybe it's breast cancer patients, but they have different types of breast cancer. Maybe you'd like to compare variation in particular genes in different types of breast cancer, or maybe you might want to compare samples by their age range, or their survival, or their treatment type. You can basically define these groups for yourself. And then you can look at different properties of annotations, such as variant frequency in particular genes, or in fact, you can select any annotation in an annotator that you've selected and compare and see how it differs between the different groups you've selected. So this is something we're still working on, and it will be released sometime next year. Okay, that's pretty much all I have to say. But I would really hope that after this talk, I've stirred some interest on your part in the tool. And here's just an overview of some ways you might access the tool. So you can go to Run OpenCRAVAT and run OpenCRAVAT on the web. You can install a Docker container on your own system. If you're a Python user, you can easily install with pip, and you can also use Windows or Mac OS native installers and install it that way. And last of all, just want to acknowledge our team. We've got a team of software developers and a bioinformatician. We are currently hiring. We're going to be adding a new bioinformatician to our team, and we actually have a brand new software developer who is not shown on the slide. And I want to acknowledge the ICM at Hopkins and Johns Hopkins itself for employing me for 17 years now, and also to NCI and ITCR, who have given us wonderful support over the years with these grants. So here again is our landing page. And with that, I am really glad to take any questions. Thank you. Thank you for listening.
Oh, thank you so much, Rachel. This has been great. If you can see in the chat, there is a number of questions that came in together. If not, I can read this for you.
Now, let me see here. I think I'm able to, here, pop out. Oh, this is interesting. Okay, for the computational predictors, could you answer, was the variant used in the training data set? Was functional evidence used for the variant? Was hotspot data used for the variant? Was protein data used? Was conservation used? Was population frequency used? So that's very interesting. So this would be a drill down into the computational predictors to basically provide, this is kind of like metadata for a computational predictor that is a very, very interesting idea. Thank you, Dimitri. We had not thought about this, but I think this would be incredibly useful. It's also particularly useful for people who don't want to double dip, like if they're trying to follow the ACMG guidelines, right? And they want to use population frequency as a piece of evidence and also use a computational predictor. You couldn't use a computational predictor that had used population frequency. And of course, if the variant that you're interested in was used in the training set, well, it's not a real prediction. It's just telling you what was in the training set. Well, this is a terrific idea. I will definitely bring this to our team, and we will get this in the queue. I think this is a great project.
Wonderful. Okay, there's one more there. Can we use it for annotation of whole genome sequencing data too?
Oh, absolutely. I mean, it's not specialized for whole exome, and it's not specialized for missense. For some reason, somebody told me recently that they thought it was for missense mutations, but it handles all kinds of small scale variants, coding and non-coding, you know, insertions, deletions, complex small variants, you know, in frame and out of frame. So it really handles, and soon it's going to be handling large scale structural variants. But absolutely, we would really encourage you using it for whole genome sequencing.
More there. What about copy number variants? Does it call CNVs and annotate them?
It does not call variants at all. It assumes that the input is the output of a variant caller. So for, you know, for mutations or germline variants, you wouldn't put in your BAM file. You would have used a variant caller and you'd put in something like your VCF file. So right now it is not annotating copy number variants, but it's coming. It is coming all in with the structural variant thrust that we're involved in right now, because we think copy number variants are just a type of structural variant. So yes, I think we're answering that. I am going to copy Dimitri's question here and get it into a buffer so I can bring it to our team.
Wonderful. Are there any other questions? You can either unmute and ask, or here's one in the chat. Go ahead. Is there any limit for the number of samples in cohort analysis?
Well, I mean, in theory, no, but I mean, in practice, I would think, so I would say two things. It's not just the number of samples. In fact, it's also the number of annotators you use and the number of variants in each sample. I think the most rate limiting thing is the number of annotators you use. So for example, if you put in 500 samples and you asked for, you know, 5 annotations in each one, I think it would be completely fine. If you used 80 annotators in each one, I think you probably it would be quite slow with 500 samples. So maybe a practical, again, so I hope that answers the question. If you're thinking of using a really, really large number of annotators, I would limit to just a few hundred samples. Certainly jobs with 30 to 50 samples, it can handle very well and was designed for that. For jobs with a really large number of samples, I would suggest we are going to be including this VCF to VCF pipeline that I talked about that we set up for DNA Nexus. That is something that could be able to handle 1 million samples. And we're going to make that available soon in just the standard OpenCRAVAT. Thank you very much for your questions.
Wonderful. If there are no other questions in the chat here, we can go ahead and wrap up. I'm just giving everyone one more minute if anyone has a lingering question. And of course, as always, if If anyone has any questions, you can feel free to email myself, Kayla Strauss, or email CBIT Informatics and IT Training. So thank you again to Dr. Rachel Karchin for your time today. Thank you all for attending and for your wonderful questions. And thank you for this overview. This was very helpful. I will be sending out the recording. And Rachel, if you don't mind sending me your slides and information, and I will make sure to get that out to everyone. So thank you all again.
All right, I'm glad to send the slides.
Perfect.
Thanks for
Thank you,
coming.
Rachel. Thank you,
Bye.
everyone. Bye.